Good morning, student. We will continue the carbohydrate metabolism, and today's topic is gluconeogenesis pathway. First, we will see definition. Synthesis of glucose from non-carbohydrate substances is called as gluconeogenesis. Here, the word non-carbohydrate substances is very important. Then, site. This pathway occurs partly in the cytoplasm and partly in the mitochondria of liver cells mainly and in renal cortex. Now come for the substrate for gluconeogenesis. That means the non-carbohydrate substances required for gluconeogenesis. These are gluconic acid, amino acids, lactic acid, glycerol and propionic acid or propionyl CoA. Sometimes a 2-mark question is asked, enumerate the 4 substrate of gluconeogenesis. So these are the substrate required for gluconeogenesis. In the gluconeogenesis, the majority of the steps are reversal of glycolysis, except these 3 steps, where pyruvate is converted into phosphoenol pyruvate, fructose 1,6-bisphosphate is converted into fructose 6-phosphate and glucose 6-phosphate is converted into glucose. Except these 3 steps, the remaining steps of gluconeogenesis are just reversal of glycolysis, and we have already studied the process of pathway of glycolysis. Therefore, we will not study the remaining steps. We will study only these 3 irreversible steps of gluconeogenesis. So these are the 3 steps: conversion of pyruvate to phosphoenol pyruvate. Second is conversion of fructose 1,6-bisphosphate to fructose 6-phosphate, and the third irreversible step is conversion of glucose 6-phosphate to glucose. So we will study only these 3 steps here. So this is the first step of gluconeogenesis, in which pyruvate is converted into phosphoenol pyruvate. The pathway of gluconeogenesis starts from mitochondria, where pyruvate is available. This pyruvate is carboxylated to form 4-carbon oxaloacetate with the help of enzyme pyruvate carboxylase, which requires coenzyme biotin. To join the 2 substrates, that is pyruvate and carbon dioxide, the enzyme requires energy which is obtained from hydrolysis of ATP. So this enzyme belongs to synthetase class, means it joins 2 substrates with the help of energy. Now question is, why this step is required? This is because pyruvate is impermeable by inner mitochondrial membrane. Only this step occurs in the mitochondria and the remaining steps occurs in the cytoplasm. Therefore, pyruvate has to enter into the cytosol or cytoplasm. But inner mitochondrial membrane is impermeable for pyruvic acid. Therefore, it gets converted into oxaloacetate. But inner mitochondrial membrane is also impermeable to oxaloacetate. Hence, this oxaloacetate is reduced to form malate or malic acid in the presence of enzyme mitochondrial malate dehydrogenase. Here, reduction occurs by addition of hydrogen atoms, which are supplied by reduced NADH. Now this malate is permeable to inner mitochondrial membrane, therefore it travels through the inner mitochondrial membrane and enters into the cytoplasmic compartment. In the cytoplasmic compartment, the reverse reaction takes place. The malic acid or malate is oxidized to oxaloacetate with the help of enzyme cytoplasmic malate dehydrogenase. So malate dehydrogenase will catalyze the conversion of oxaloacetate to malate as well as malate to oxaloacetate. Here oxidation occurs and 2 hydrogen atoms are removed from malate, which are accepted by NAD to form NADH, and oxaloacetate is formed. Now this oxaloacetate is broken down into phosphoenol pyruvate with the help of enzyme PEPCK, that means phosphoenol pyruvate carboxykinase. Now this enzyme breaks the oxaloacetate into 3 carbon phosphoenol pyruvate by removal of 1 carbon in the form of carbon dioxide. For this reaction also 1 GTP is required as a energy source. To regenerate a glucose molecule, we require 2 pyruvate molecule should be converted into 2 phosphoenol pyruvate molecule, because in the glycolysis we have studied that when 1 molecule of glucose is oxidized in glycolysis, 2 pyruvate are formed. So here also, if we want to regenerate the glucose molecule, the 2 pyruvate molecules should be converted into 2 phosphoenol pyruvates. So the energy requirement which I have shown here, it is doubled in 2 reactions. Now the second irreversible step of glycolysis or second step of gluconeogenesis is conversion of fructose 1,6-bisphosphate to fructose 6-phosphate with the help of enzyme fructose 1,6-bisphosphatase, which removes a phosphate bond from the first carbon atom of fructose 1,6-bisphosphate by the process of hydrolysis, and the phosphate is removed as inorganic phosphate and fructose 6-phosphate is formed. So remember the enzyme fructose 1,6-bisphosphatase. The third step of gluconeogenesis is conversion of glucose 6-phosphate to glucose with the help of enzyme glucose 6-phosphatase, which again causes hydrolysis of the ester glucose 6-phosphate to free glucose with the help of water, and again the phosphate bond is removed in the form of inorganic phosphate. Now importance of this glucose 6-phosphatase is it is mainly found in the liver. So free glucose can only be regenerated in the liver, but small quantity of this enzyme is also found in renal cortex. So this is about the gluconeogenesis pathway. Here we have to describe only about the 3 irreversible steps of glycolysis. Complete pathway need not to be described when the question on gluconeogenesis is asked. Now come to the next topic, energetics of gluconeogenesis. 4 ATP molecules are required for synthesis of 1 molecule of glucose. 2 at each step, that is the step catalyzed by the enzyme pyruvate carboxylase and phosphoenol pyruvate carboxykinase. 2 pyruvate molecules are required for synthesis of 1 molecule of glucose. Once again, see this step where energy is required. The first energy requirement step in gluconeogenesis is conversion of pyruvate to oxaloacetate in mitochondria, where ATP is required by the enzyme pyruvate carboxylase to join the pyruvate and carbon dioxide. As we are using 2 pyruvate to form 1 glucose molecule, the energy requirement in this step is 2 ATPs. And the second step where energy required is in the cytoplasmic compartment, where oxaloacetate is converted into phosphoenol pyruvate. The enzyme is phosphoenol pyruvate carboxykinase. As again here 2 phosphoenol pyruvate molecules are required. Here also 2 GTPs should be utilized in case of gluconeogenesis. So the 4 ATPs are required for the synthesis of 1 molecule of glucose at these 2 steps. Now come to the regulation of gluconeogenesis. First is hormonal regulation. Glucocorticoids, thyroid, glucagon and adrenaline stimulates the process of gluconeogenesis, whereas insulin inhibits the process of gluconeogenesis. Gluconeogenesis mainly occurs in fasting state when glucose is not available in the cell. Then cell try to synthesize glucose molecule from non-carbohydrate substances. So in fasting state, the hormone present in our body are glucocorticoids, thyroid, glucagon and adrenaline. So these hormones stimulates this pathway. Insulin is synthesized in post-meal state. At this state, the glucose level is high in the body due to absorption from gastrointestinal tract. So body does not want new glucose from gluconeogenesis pathway. Hence, insulin inhibits the process of gluconeogenesis. Next is allosteric regulation. The two enzymes of gluconeogenesis are very important. These are pyruvate carboxylase and fructose 1,6-bisphosphatase. These are called allosteric enzymes because they have two different sites, one for active site and other for modifier. That site is called as allosteric site. So these enzymes are stimulated or inhibited by the modifier. If the modifier binds at the allosteric site, it stimulates the enzyme. If the modifier is inhibitor, then the enzyme is inhibited. So these are the allosteric stimulators and allosteric inhibitors of gluconeogenesis. First is pyruvate carboxylase, acetyl-CoA. When the concentration of acetyl-CoA increases inside the mitochondria, it indicates that the patient is in fasting state. This is because in the fasting state, the fatty acids are oxidized to form large amount of acetyl-CoA. So when the concentration of acetyl-CoA increases, it stimulates the enzyme pyruvate carboxylase and in turn the complete pathway of gluconeogenesis is stimulated. If the concentration of ADP is high, then it acts as an inhibitor inside the cell. The concentration of ADP high indicates the ATP concentration of the cell is low. That means there is low ATP inside the cell, and just we have seen 4 ATP molecules are required for synthesis of one molecule of glucose. So if the ATP level is low or ADP level is high, then that ADP act as a inhibitor of gluconeogenesis, or cell cannot afford the gluconeogenesis pathway when ATP level is low. Now come to the next enzyme, fructose 1,6-bisphosphatase. When the concentration of citric acid inside the cytoplasmic compartment increases in fasting state, it stimulates the enzyme fructose 1,6-bisphosphatase, which in turn stimulates the pathway of gluconeogenesis. Presence of high ATP also act as a allosteric stimulator because energy requirement is very important for gluconeogenesis, because 4 ATPs are required for synthesis of one glucose molecule. So presence of ATP inside the cell is an important factor of gluconeogenesis. Now come to allosteric inhibitors of the enzyme. AMP act as a allosteric inhibitor. Again the reason is same. Presence of AMP inside the cell indicates that the ATP concentration of that cell is low and cell cannot afford the process of gluconeogenesis. Therefore AMP inhibits the enzyme. The second inhibitor is fructose 2,6-bisphosphate. So this is the fructose 2,6-bisphosphate. When the concentration of fructose 6-phosphate is high, then the fructose 6-phosphate is converted into fructose 2,6-bisphosphate with the help of enzyme phosphokinase 2. high concentration of fructose 6 phosphate indicates that the glucose concentration of the cell is also high. And this fructose 6 bisphosphate is generally formed after meal or post meal state when glucose concentration is high. Therefore, the fructose 2,6 bisphosphate inhibits the enzyme and act as a allosteric inhibitor of this enzyme. So this is about the allosteric stimulator and inhibitor of gluconeogenesis. Now come to the clinical part, disorders related to gluconeogenesis. First is deficiency of enzyme pyruvate carboxylase. It is an inborn error, that means present since birth. It is found in 1 in 25,000 live birth. The patient is presented with mental retardation. In these patients, the process of gluconeogenesis is inhibited or taking place at slow rate. When that pyruvate get accumulated, as it is not utilized for gluconeogenesis due to deficiency of the enzyme pyruvate carboxylase, the pyruvate get converted into lactate, and that lactate deposits in the cell leading to lactic acidosis. So this is pyruvate carboxylase deficiency. Second clinical disorders which is very commonly seen in clinical practice is alcohol or ethanol inhibits the process of gluconeogenesis in alcoholic patients and it leads to hypoglycemia. To know the mechanism how alcohol causes hypoglycemia, you should know the ethanol metabolism. Ethanol is converted into acetaldehyde and acetaldehyde is converted to acetic acid. Both these steps occurs in the cytoplasmic compartment of the cell. So enzyme required for these steps are alcohol dehydrogenase and aldehyde dehydrogenase. So if you see in these steps, in each step NAD is converted into NADH. So when one ethanol is converted into acetic acid, two NAD molecules, one at each step, are utilized and they get converted into NADH. Therefore, there is deficiency of NAD inside the cytoplasmic compartment of the cell because the metabolism of ethanol takes place in the cytoplasmic compartment of the cell. In short, once again I will repeat, to metabolize one ethanol molecule to acetic acid, two NAD are utilized. Now come to the first step of gluconeogenesis. So this is the first step of gluconeogenesis, and if you see in the cytoplasmic compartment, when malate is converted into oxaloacetate by the enzyme malate dehydrogenase, this step also requires coenzyme NAD. And a patient who have consumed ethanol in large quantity, he has utilized the NAD of the cytoplasmic compartment and hence NAD is not available in the cytoplasmic compartment. Therefore, the conversion of malate to oxaloacetate does not take place and the gluconeogenesis pathway is inhibited. So this is the mechanism of hypoglycemia in alcoholic patients. It is a very common condition and the patient presents with coma or semi-coma. Second clinical condition related to alcohol is ethanol causes lactic acidosis. The mechanism is same. When ethanol is metabolized in the cytoplasmic compartment of the cell, two NAD molecules are utilized and there is deficiency of NAD molecules. NADH level is high, but NAD, oxidized form, of NAD are low in the cytoplasmic compartment. Therefore, cell try to regenerate the NAD molecule. And how it will regenerate? It will regenerate the NAD molecule by converting the pyruvic acid or pyruvate into lactate. So in this step, which is catalyzed by the enzyme lactate dehydrogenase, NADH is converted into NAD. So to regenerate the NAD, the cell will convert more and more pyruvate into lactate. Therefore, lactate is accumulated inside the cell, leading to lactic acidosis. So these are the clinical diseases related to gluconeogenesis. Now come to significance of gluconeogenesis. Only liver can form glucose, free glucose, because the enzyme glucose 6-phosphatase is present mainly in the liver. So free glucose is formed only in the liver, and the liver can only replenish the blood glucose in fasting state. So this is that step. Glucose 6-phosphatase is convert the glucose 6-phosphate into free glucose. So free glucose is formed in liver, and from liver it enters into the blood and maintains the blood glucose level. And liver is responsible for maintaining the blood glucose level in fasting state. During starvation or fasting state, the blood glucose level try to decrease. Up to 12 to 18 hours, the blood glucose level is maintained by the process of glycogenolysis, which is breakdown of glycogen to glucose. But after that, the glycogen stores of the body get depleted. Now, the gluconeogenesis is the only pathway which is responsible to maintain the blood glucose level. On prolonged starvation, the gluconeogenesis get speed up, and it utilizes amino acids which are obtained from protein catabolism as a source of to form the glucose, or it utilizes the amino acid as a substrate for gluconeogenesis. So this is about gluconeogenesis pathway, its regulation, clinical disorders and significance. Thank you very much.